Okay guys, so last class we were working on these notes, which were the light reaction notes. Take a minute, find these. You need the second page today. You may want to pause the video until they have them ready. So today we're going to look at the light independent reaction, which is also known as the dark reaction, and also the Calvin cycle. Dark reaction, Calvin cycle. They're all three the same. They are independent of light. We do not need sunlight at this point. So in our chloroplast, remember this chloroplast diagram from last class? We were looking at the light-dependent reactions in the granum, thylakoid disc. Now we're moving over to this side of the diagram in this fluid-filled space that we call the stroma. And this is going to be where our focus is now. So when we look at this question, chloroplast location, that is stroma. Again, don't get it confused with stomata or stoma. That's the holes on the other side of the leaf. This is in the chloroplast. Dark reactions can occur in light or dark. So it can occur during the daytime when there's light, or it can occur when there is no light. It is all driven by the ATP that comes from the light reaction, like we saw in our last notes. It is going to join our other reactant, carbon dioxide, with those hydrogen ions that are pulled apart from the water when it is broken apart in the light reaction, the photolysis of water. So I'm going to put here from the light reaction. And ultimately, the CO2 and the H's will rearrange to form our glucose molecule, which is the C6H12O6, and our final product of photosynthesis. The actual product is not glucose, so the very first step or first product we get from the light independent reaction is not actually glucose. It is instead G3P or PGAL. PGAL is an older terminology, and that stands for glyceraldehyde 3-phosphate. So G3P. That might help to put it where you can see it. There we go. There are three phases to the Calvin cycle. The first phase is carbon fixation. The second phase is a reduction phase. And the third phase is a regeneration or a rearrangement. So now if we look at the diagram below, this is a simplified version of one that we're going to do in a minute, but this is going to start out with this molecule, which is our reactant carbon dioxide. There are actually three of these that go into the Calvin cycle, and it is going to enter in this kind of first triangle at the top here, phase 1, which is carbon fixation. And we'll get more specific about what that means in the other diagram where we have more room. But then if we move down in our diagram here to the next step, phase 2. Phase 2 is now in this area. So we'll put phase 2 down here. Nope. Okay. And this is our reduction phase. Notice here's our energy molecule. So here's that ATP coming from the light reaction. It's going to be used in the reduction phase, and that energy is going to be used to make these new molecules, and we're going to end up with our ADP coming out of that, plus that free-floating phosphate that was broken off to give energy to this reaction. And here, this is our NADPH molecule that also comes from the thylakoids of the light reaction. It's going to be used, it's going to be broken apart, and it is going to be regenerated into NADP+, and free-floating inorganic phosphates and hydrogen ions. As a result of phase 2, we get this molecule right here, which is the molecule that we are looking for at this point. This is our G3P molecule, our glyceraldehyde 3-phosphate. And there should be 6 of these by the time you get to this step in the phase 2 reduction stage. And here, coming out of the cycle, will be 1 of the G3P or P-gals. This 1 goes on to make glucose. So that is where our glucose eventually comes from. The other 5 of these continue on. So this right here would be 5 G3Ps, which are left. They're going to continue over to phase 3, which is regeneration or rearrangement. So we take these G3P molecules with more ATP, more energy is needed. So energy from ATP breaks back down ADP plus a free floating phosphate. These carbon molecules get rearranged, hydrogen molecules get rearranged to form this molecule here. Notice we go from a 1, 2, 3 carbon to a 1, 2, 3, 4, 5 carbon molecule, and this molecule is called Ruby P for short. It's ribulose biphosphate, but we call it Ruby P. Ruby P joins with the CO2 in the carbon fixation to begin the cycle all over again. So that is our Calvin cycle, named after the person that discovered it. That is where we get ultimately our final product for photosynthesis, glucose. To finish the notes, if we look down at the bottom here, we have some plants and some organisms that take alternative pathways to get to the different products. So we have C3 plants, which only use the Calvin cycle. We have C4 plants, which are 4 carbon compounds. And that's found in the Calvin cycle as well. And then we have CAM plants. CAM plants are commonly known as succulents. Those are the ones that you see all over now that are really small, and you sometimes call them air plants, or they are your cactuses and those types of plants that don't need just a lot of water at one time. And so their stomata are open at night to reduce water loss. So if they're found in the desert area, which would be common, then they want to retain as much water as possible, and they don't open during the daytime where there's sunlight. They open at night where it's cooler and not as much water to be lost through evaporation. Then we have factors affecting the rate of photosynthesis. The rate, how quickly, how slowly photosynthesis may occur. Those factors are the intensity of light, concentration, amount of water, and carbon dioxide. This reaction will not go until there are 3 CO2 molecules, and then it will enter this and start the Calvin cycle. So it does depend on the amount. And temperature. Temperature is another defining factor in how quickly or how slowly photosynthesis takes place. Okay, so that's the last of those notes. Take a minute. Find your diagram. This diagram is what we're looking for. So take a minute, pull that out,